Okay, so now I think we can start. Let's switch to English. So welcome everyone who joined this webinar today. So we are so happy to have here with us a former student from USP, from the Ribeirão Preto Medical School, Dr. Renan Carvalho. So thank you so much for accepting our invitation, Dr. Renan. Just a brief introduction before he starts. Dr. Carvalho has a bachelor from the Universidade Federal de Juiz de Fora, with an internship at the Universidade do Porto, Portugal, and at the Institut Pasteur in Paris, France. So then he moved to Ribeirão Preto to perform his PhD at the Professor Dario Zamboni's lab, where he was also a postdoctoral fellow. And now he's a postdoctoral fellow in the Laboratory of Lymphocyte Dynamics, coordinated by Professor Gabriel Victoria, another good friend of ours here in São Paulo. So at the Rockefeller University in New York. So, Renan, we are so glad of having you here today with us. Thank you so much again. For the audience, we are open to questions to Dr. Renan at the end of the presentation. So feel free to open your mics or raise your hands. You can put the questions in the chat as well as you want. So, Renan, the floor is yours. Thank you so much.
Thank you, Luisa. Yeah, first of all, I'd like to thank for the invitation. It's a big pleasure for me to be here, talking to maybe two of the most important scientific hubs in Brazil, Federal University of Rio and also University of São Paulo, which was, like she said, also my home for several years. So it's a big pleasure. I'm very glad for the invitation and the opportunity to be presenting here my work to you all. So maybe, can you see my screen? All good?
Yes, now it's full screen mode.
Okay. So today I'm gonna show you, so like Luisa said, my name is Renan. I'm a third-year postdoctoral fellow at the Laboratory of Lymphocyte Dynamics here at the Rockefeller University in New York, where I work with Professor Gabriel Victoria. And I'm transitioning now to my fourth year, beginning of my fourth year of postdoc. Now I'm a CRI, Cancer Research Institute postdoctoral fellow. Previously, I was a Human Frontier fellow for these initial 3 years. And today I'm gonna walk you through this study that we just concluded. It was published like a few months ago in the beginning of this year, where we explored and characterized better these structures, the long-lived germinal centers that are primed by influenza virus. And we identified this phenomenon of clonal replacement within these structures. And I'm gonna walk you through the data and the hypothesis and how we managed to get what we did. So initially, I'm gonna split this talk. Initially, I'm gonna tell you a little bit about this basic thing about B cell activation and germinal center formation. So this is a very classic, I'm sure that you all immunologists are very familiar with the ABAS workbook. And I like this figure because it summarizes very well the initial steps of B cell activation. As we know, whenever we have like antigen being presented, when you get in touch with a pathogen, with an infection or an immunogen, you get your dendritic cells to travel to the lymph nodes, where they will prime T cells, and this leads to T cell activation. Of course, I'm not going to talk about this during this talk, but it's important to contextualize these events, of course. But also, I would like to pinpoint here the importance of the B cells, and this, of course, happens in secondary lymphoid organs, when among the follicles of lymph nodes and spleen and so forth, where B cells can capture this antigen that is trained to these lymph nodes, and they can present to the T cell and initiate a TB interaction. So here in the border of the TB zone, we have this interaction, and this can lead to the activation of B cells. And one of the fates that T cells can assume is the differentiation into plasmablasts, right? So these are the extrafollicular plasmablasts. They are not mutated. They just class switch and they become plasmablasts, and they secrete antibodies locally for a short period of time, and they're presumably dead after a few days, still in the lymph node. But another fate that these cells can assume once this interaction happens is that they can become germinal center cells. And I'd like to emphasize here that these events are not mutually exclusive. So for example, whenever this contact happens, you can have a proliferation of these B cells, and some of them will become plasmablasts, and some of them will lead and transform into a germinal center cell and initiate the germinal center reaction. And it's very important also to mention that this is just a piece of data from a paper that we published in the lab a couple of years ago from Luca Mezzing and Ariane Schippers in the lab. And here you can appreciate that although, as you know, we have up to 10 to the 14 combinations of BCRs in the B cell repertoire in the body, we can appreciate here that only 100 clones are normally initiating the germinal center reaction. So among a pool of an infinite pool of B cells, only those BCRs that can recognize the antigen and present to the T cells among those among that B cell pool, they can actually join the germinal center reaction and initiate it. So here it was in the context of chicken gamma globulin, which is an immunogen when administered with alum, but it also happens during infections and all other kind of immunizations that we know. So just to emphasize that we really select for those clones that have a certain affinity and are capable of presenting these cells to T cells and initiate all of this. But now I'm gonna give a zoom in what really matters to this talk, which is the germinal center reaction. And we know if I zoom on here, this is a very well-known process of B cell activation, and this is the ultimate outcome of this germinal center reaction is to generate high affinity B cells that can neutralize antigens with a very high affinity. And here it seems to be a very complicated figure from a review also published from the lab, but I'd like to make it very didactic for you. So here we can distinguish the germinal center between two zones, the light zone and the dark zone. And the dark zone is where the B cells undergo rounds of proliferation and mutation. So there is this enzyme here, AID, which promotes class switching and the mutations, random mutations in the BCR genes that will alter the affinity of these cells because it's going to change some amino acids eventually in the BCR structure, and that will lead eventually to a high affinity of the B cell. And once these cells undergo this proliferation in the dark zone, they can get back to the light zone, and here they're going to be tested and selected by TFHs, which are the T follicular helper cells. And this process will lead to the selection of the B cells and differentiation eventually into plasma cells and the memory B cells, which are the two different fates that surviving B cells in the light zone can assume. So either they will become plasma cells and presumably with a higher affinity, or if they have a somewhat lower affinity, they upregulate other genes and become and get exported from the germinal centers as a memory B cell. But like I said, the process of proliferation and mutation given by AID in the dark zone is very random. So the likelihood of a B cell to get a higher affinity for the antigen is very low, right? Which induce the majority of these B cells to be eliminated by apoptosis and get eaten by these tingible body macrophages that eliminate B cells in the light zone and also in the dark zone. It's important to mention the role of the follicular dendritic cells as well here, which are these elongated cells that have antigen in their structures, and the B cells actually capture this antigen and can present this antigen to TFHs, which lead to the selection of the B cells. So overall, this is very important for you to know that this structure indeed gets to the selection of these very high affinity cells. So among the pool of B cells that enter and proliferate in the germinal centers, the germinal center is very efficient to select the high affinity ones, and then these cells get exported as plasma cells and memory B cells. And this is very important for the end of my talk that I'm going to talk later on. Okay, and this is pretty much what I just mentioned to you all. I just wanted to emphasize that the reason why germinal centers exist to some extent, if we can say that, is to induce this process of affinity maturation, which was described by Hermann Eisen decades ago, where he described that the mean affinity of serum antibodies goes on, takes off with time. And that happens because these B cells are able to undergo these rapid cycles of mutation and efficient selection of the ones with high affinity. And it's very important to mention that the longer that a B cell stays in the germinal center, one would assume that the more likely they are to produce high affinity antibodies. And nowhere it's more true than in the case of HIV, for example. So in HIV, we know, for example, those BNAbs, the broadly neutralizing antibodies produced by certain B cells that some people have. They have those B cells have almost 100 mutations in the BCI gene to enable the broadly neutralizing effect, which assumes that those cells stayed a long time in the germinal center. So presumably the HIV induced germinal centers could be long-lived, although this is still speculative. And overall, that's it. The germinal center is this process, a Darwinian process of evolution, where the high affinity BCRs are selected. So those reacting and producing antibodies against the antigen with a very, very high affinity get selected to survive and get exported as either memory or plasma cells. And with that, I just want to mention, and this is the key point where we transition to the talk, which is the duration of the germinal centers, right? So the way we know the germinal centers as it is this process where you have like an immunizing agent, let's say OVA plus alum or an adjuvant, and the GC goes on and reaches a peak a few weeks after the immunization or infection, and then mechanisms related to TFRs and FOXP3 regulatory cells emerge at the end stage of these germinal centers and is responsible for the shutdown of these structures. So there is like an expansion phase of the germinal center, and then you have this contraction phase, and that sort of terminates the germinal center reaction. The GC can also be prolonged by, for example, a more potent adjuvant. If you give alum or allyhydrogel, you can extend the germinal center duration to a few weeks more. But what is really striking to observe is the case of certain viral infections, for example, influenza and VSV. And here I compile a series of papers describing this process. Not only viral infections, but we see also in the case of plasmodium infection in mice, germinal centers that last way more than canonical germinal centers do. So for example, plasmodium, flu, VSV, we also see LCMV and hepatitis B virus, the model that Charles Rice used here in his lab at Rockefeller. They induce these structures that go on for a long, long, long time. And those are the so-called long-lived germinal centers. Recently, Teddy Eldo, that's now a principal investigator at Genentech, he described when he was doing his postdoc in Jayanta Shaburi's lab here in MSK, these long-lived germinal centers induced by influenza. And more recently, we have Ali Aleberi's group in WashU demonstrating that the mRNA from spike used in the vaccines that we have against COVID, they also induce long-lasting germinal centers in humans. And also now, Shane Crotty's group published a very elegant paper in Nature demonstrating that immunization with the HIV envelope protein in monkeys can lead also to these germinal centers that last for a very long time. So here's the part that we start our study, because we basically asked using flu, which is our favorite viral infection model that we use in mice, what are the characteristics of these germinal centers, right? What happens to them? So we know that the canonical ones, like I mentioned to you before, everything that I told you about germinal centers before was studied and understood in the context of these canonical germinal centers induced by immunization or infection. But what are the characteristics of these germinal centers that last for a long time? Are they are they behaving as they should? Are they having differential properties? So how do they look like? And for that, the first experiment that we performed was basically what Teddy Eldo described in his paper in the reports 2 years ago, which is this kinetics experiment using flu in wild-type mice. So infected with flu, and we looked at different time points after infection. And what we see is basically the size of the germinal centers in the mediastinal node. So the node that stays between the heart and the lung that drains the infection. And we can see that these germinal centers by week 2, they are very, very big. They get even bigger at 4 weeks post-infection, and they start to decay over time until 24 weeks post-infection. And this is quantified here with a lot of different mice in each of these time points. And I just wanted to mention that 24 weeks, we still have bigger GCs compared to age-matched controls that are non-infected. So these GCs indeed persist for a long time. And here, just for you to understand, this is how we characterize FASI CD95 for those that are not familiar with the GC gating. So among the B cells, those that are FAS positive and CD38 low are the ones in the germinal center. So that's why we gate them like this. And among these germinal center cells, we can also see the frequency of them that are binding to flu antigen in the case of hemagglutinin, which is one of the main immunogens, one of the main proteins that our immune system recognizes from flu. So if we evaluate the percentage of the frequency of binders among the germinal centers over time, we see that they also decay, but they still, for example, 4 months after infection. It's really a lot. If you think that a mouse lives for 2 years on average, that these GCs are persisting up to 6 months after infection. So it's basically 25% of a mouse lifespan, which is a lot, right? It's basically, if we translate that to humans, that would be considering like, if a person normally lives 80 years, let's say on average, this should be like GCs persisting for 20 years in humans, which is like a lot if we convert to humans. So indeed, these are phenomenal GCs that for some reason are persisting for a long time. And so the question that is raised here is that, okay, so we are describing this, this is also has been shown by other groups, like I mentioned previously, but what are the mutational characteristics of these GCs over time? So one would assume that the longer a GC B cell stays in the germinal center, the higher the number of mutations they would accrue. So we decided to sequence the germinal center cells in these different time points. And here we basically sort single cells from the germinal center, and we sequence and we get the BCR sequence of them. And we can also get like characteristics like the number of mutations on them. And here, basically, each of these dots is a different cell. So we have like 2 mice here, 2 mice here, 2 mice here, 3 and 2 and 2 in these different time points. And each of these dots are a cell. And we can see that indeed, like from 2 weeks until week 12, there is a takeoff in the number of mutations. So on average, the number of mutations goes up to week 12. But what was really surprising to us is that later on, at week 16 and week 24, the average number of mutations actually go down rather than taking off forever, right? So it's not a linear curve that goes up indefinitely, but rather this is going down, especially after week 16, even though we do see some cells that are highly mutated here in these late time points, but on average, this is getting down, which was very surprising to us because this is not going necessarily in accordance with the GC as we think about it. So how to explain this? So how to understand what's going on in these GCs? So this is a comment, a paper that was published, Victoria Gabriel published with Hugo Moquet, that's now PI in Institut Pasteur in Paris, in Cold Spring Harbor. And they basically postulated that these long-lived GCs, there are 2 possibilities for them. Either they were triggered by antigen, and the clones that seeds the GC reaction are intrinsically long-lived and they persist up to, let's say, 6 months after infection, or they are continuously seeded by incoming B cells. So it's not the same clones that seeded the reaction are not those that remain to the end. You can still have some here, but you have like recruitment of B cells over time to these germinal centers. So what what would be the most likely hypothesis here? So we decided to address this. And for that, we took advantage of our favorite fate mapper mouse model, which is the S1PR2 CREAT2 Rosa tomato. So basically, S1PR2 is a marker of germinal center B cells, and it's linked to a inducible tamoxifen promoter in this gene. And we have this stop flux with the tomato protein, the Rosa 26, which is a ubiquitously expressed gene in every cell. So the way that this mouse works is that when we pulse this mouse with tamoxifen, we put a red tag on the germinal center cells. So the germinal center cells that are formed on that moment, they start to express tomato. And this is a property that they will do forever since it's linked to the Rosa 26 locus. Whenever these cells are stepped upon tamoxifen administration, they will be red forever, those cells that are in the germinal center, and also their progeny, the memory cells and the plasma cells that get exported. So we infected with flu these mice at week 0, and 2 weeks later, in the peak of the germinal center reaction, as I showed you before, so these are the persistent germinal centers in the tomato mouse, just like I showed you in the wild type before. And when we pulse with tamoxifen at week 2, we are basically fate mapping. Fate mapping means putting a timestamp on all these germinal center cells. And we performed flow cytometry from week 3 to week 24. And the result was very, very also surprising to us to some extent, because we see that at week 3, like a week after our tamoxifen pulse, we have almost 100% of our germinal center cells fate mapped. So they are expressing the tomato protein. Which was very intriguing to see is that over time, those cells that seeded the reaction, that initiated the germinal center reaction against flu, they're actually going down and getting replaced by invader cells. So those that see the reaction, and this is a very important point for you to keep in mind, that I'm gonna call these cells that are responding to flu and initiated the germinal center reaction, the founder cells, those that really triggered the response against flu. And those cells that replace the red ones that are going away over time, I'll be calling the invaders, the dark cells, the invaders, which are the cells that are replacing these red founder cells. So it's a very important, this is perhaps one of the main piece of data from this study, because it shows that the clonal replacement, so the replacement of the cells that triggered the germinal center, is happening to a very high extent, especially after week 12, that we see that almost 50% of the GCs, the GCB cells that see the reaction are gone. So we also sequence these cells, and we can appreciate from these clonal pies, as we call them, that when you look at week 3, you have this very low number of invader cells, and the majority of the cells are the founder ones. But as time goes on, we see that the fraction of the invaders actually go up with time. And at week 16, for example, we barely see any red cells. So those cells that were responding to flu initially here to the PR8 strain, they're not there anymore. And if you look at week 24, there almost everything is gone, right? You have very, very few cells that are red. And another very important point to highlight here is that so each slice of this pie is a different clone. So for example, if we look at week 8 after infection, we see that there is this clone that is basically 25% of the germinal center. So this is a clone that underwent a massive clonal burst, and their progeny is taking over the GC. And still, this is a very polyclonal compartment, because we have like different clones in the beginning, right? Then you select for a few of them here, and it gets even more scarce over time. And this is also a property from the invaders compartment, where we see that there is this high polyclonal compartment, although we can see some clones that can actually expand even long after infection. So we also now have this resolution, since we fate mapped and we have this tomato protein on the founder cells. Now we can actually distinguish the number of mutations, because before we were looking at the total germinal center cells, so we weren't sure who the invaders and the founders were. But with this new resolution, we can actually track independently the red founder cells and the invader ones. And if we look only at the red cells, the founder ones, we can now see that they take off over time, and at 24 weeks after infection, the cells that seeded the reaction are actually very highly mutated. So you see the bulk of them have an average number of mutations that's above 20, which is quite spectacular considering the very low frequency of these cells at this time point. And the invader cells can also go up to a certain extent, but lower compared to the founder ones in number of mutations in all the time points. But they are the ones that were pushing back the number of mutations at these late time points. So now we see that, both of them are evolving, but actually the founder cells that stay, they are very highly mutated and they could be doing something useful against flu. So overall, this piece of data suggests that the long-lived germinal centers are sustained by the turnover of the B cell clones within a persistent germinal center B cell population. And now we went to track the origin of these invader germinal center cells. And for that, we took advantage of this parabiosis experiment that we do a lot here in the lab. In this system, what we did was to take advantage of these distinct allelic markers, which are the CD45.2 and .1. And what we did was to infect only the .2 mouse, which is this one, and we induced the germinal center reaction in this mouse. And 4 weeks after infection, we performed the parabiosis experiment. And we left these mice connected for additional 4 weeks. And at 8 weeks, we sectioned both of them and analyzed by flow cytometry the invasion of these cells. And it's important to mention that flu is, in this model, in mice, it's supposed to be cleared from the body after a week of infection, right? So at 4 weeks, when these mice are connected, there is no residual quantity of virus anymore in the body, presumably. And the .1 mouse is a naive one, and it has never been seen flu before. So we presume that whenever we see cells from this mouse invading the germinal center of this one, we would assume that their origin is actually naive rather than memory or any other. So here is the result of this experiment. And again, we gate here on the node of the infected mouse, the .2 mouse. And we have here the very big GC at this time point. And when we looked at the germinal center cells from this infected mouse, we see that there is a significant fraction of cells in the germinal center that comes from the .1 mouse, which was the parabiont. So it suggests that these cells are actually capable of invading, and that sort of sustains a naive origin of these cells to the germinal centers of this one. And here is just a control showing that there is a perfect chimerism between these two mice if we look at the total B cells, total B cell compartment in these lymph nodes. So 50% of the cells are .1 and 50% are .2. So, and indeed, like I said, these long-lived germinal centers are then sustained by naive B cell clones that are joining the germinal center reaction. And now the next question was, are these naive invaders actually forming de novo GC structures in this mouse or in the infected mouse, or are they joining the GCs that are ongoing from the infection of flu? And to address this particular point, we performed the parabiosis again, but now using our fate mapper, again, the S1PR2 tomato, that we can timestamp the germinal centers induced by flu, repeating the same experiment, but instead of connecting this mouse to .1, .2, the invaders are now AID GFP. So whenever these invaders go to the germinal center induced by flu in the other mouse, we can actually distinguish these cells because they are green, and the AID is an enzyme, like I mentioned in the introduction, that is induced in the germinal centers to induce class switching and somatic hypermutations. And here is the result of the experiment. So this is the mediastinal node. This is confocal microscopy to photon. And here we can identify the clusters of germinal centers. Here we inject IV the CD35 that labels the follicular dendritic cells. And we can see, for example, here we highlighted 4 different germinal centers that are red, right? Because we use this mouse and the germinal centers start to express tomato protein. And we can appreciate here these germinal centers 1, 2, 3, and 4. And all of them have variable degrees of invaders from the AID mouse. So for example, the germinal center number 2 and number 4 are not much invaded, while number 1 and number 3 are highly invaded by these naive AID GFP cells. And here we can quantify that by flow. This is another measure that we have to show that this experiment worked. And when we look at individual GCs from 4 different mice connected by parabiosis or pairs, we can clearly see each of these dots is a different GC. So for example, mouse 1 had like 6 GCs that were very lowly invaded, which constitute just a few GFP cells, while 2 of these GCs were highly invaded, constituting 60% of green cells. So there is this differential invasion of the germinal centers that we think is related to timing, right? If you look later on, using the same experiment at week 12 or 16, we can see that they tend to equilibrate better and the GCs tend to be more equally invaded. But it's very important to mention that we never saw germinal centers that were composed only of green cells. So this really suggests that the green cells that are invading these germinal centers are actually coming to invade the flu germinal centers from these mouse. So they are always coming to these structures that are already formed, which suggests that the clonal replacement results from the invasion of ongoing germinal center structures rather than the novel formation of new GCs. And this sort of answer the previous hypothesis, which of these should be true. So we actually show that indeed these long-lived germinal centers are primed by flu, and they are continuously seeded by incoming B cells, which are these invaders. And we also pinpoint that they should be of a naive origin. Okay, and now, given that this phenomenon is being described by us, we next wanted to understand the differences between these founder and invader cells over time. And one of the very first experiments we did was to check their transcriptional profiling by single-cell RNA-seq, SMART-seq, that we repeated the experiment, no parabiosis anymore. This is just like as compared to tomato mouse that we infect, plate mapped with tamoxifen, and we evaluated 1 week or 6 weeks after infection, weeks 3 and 8. Sorry, 3 weeks or 8 weeks after infection, 3 and 8, and we evaluated by RNA-seq. And the results actually show, so for example, this is the normal discrimination that we have in germinal center cells. So just a hint here from this experiment, when you do RNA-seq in germinal center cells, everything that you find is related to cell cycle, because that's what I mentioned in the beginning. The germinal center is a structure where B cells are cycling all the time, right? They are differentiating, they are proliferating, they're receiving signals to go back to the dark zone and differentiate. And here we can appreciate different phenotypes of B cells in the GC. We have, for example, dark zone G1, light zone G1. We have cells that are in the G2M cycle, S, G2M. So we have all kinds of cells here, those expressing MYC, which is an important proton cogene in the germinal centers, the pre-memory cells, and so forth. So we have a lot of cells. And when we try to see whether the invaders and founders cluster differently among these subsets, we actually see that they do not, right? So here we have the 3 weeks as a positive control, the yellow. These we sorted basically only founders because the germinal center at this time point is composed only of red cells, and they are all over. And when we plot the 8 weeks, the invaders that are tomato negative and the founder cells that are tomato positive, we can see that they are all over also. So we conclude from this experiment that basically we don't have difference at the transcriptional level. So all the invaders and founder cells are expressing the same content, and you can find them in different phases of the cell cycle, and they are doing what they're supposed to do. So you have no difference in terms of transcription between founders and invaders. And next, we wanted to understand better the clonal characteristics of invaders and founder cells, because we saw something interesting in our first experiment with the S1P2 tomato mouse. And we then repeated the parabiosis experiment, but we crossed the S1P2 tomato mouse to the PAGFP mouse that was used by Gabriel in his cell paper in 2010, where he can actually photoactivate germinal center cells, individual germinal centers, using this photoactivated transport fluorescent gene. And once we can incide the light in these different germinal centers, and they start to become green. So we performed this experiment, this parabiosis between the tomato, PAGFP. So basically we can now fate map and photoactivate these cells with a mouse that we can distinguish by .1, 45.1 expression that is also photoactivatable. So we infected and pulsed with tamoxifen, parabiosis, the same scheme that we did with the AID GFP mouse. But now we have a different scenario. So for example, when we look here in the germinal centers, we will find those that are less invaded, that are composed only from the tomato cells, and those that are highly invaded, composed from the cells that are .1 positive. And we can distinguish the GCs, and then we can photoactivate the single individual GCs and discriminate between them. And by micro dissection, we can actually pick these GCs individually, sort them and sequence and see their clonal characteristics. And this is pretty much what it looks like. So for example, if you get these GCs that's founder dominated, only of red cells, we can see by flow after we sort these GCs that almost everything is red indeed, expressing tomato and .2. But an invader dominated GC is actually composed of large fractions composed from CD45.2 negative cells, which imply that they're coming from a 0.1 here, while the minority are actually coming from the red, from the tomato mouse, the cells that seeded the reaction. And we also have those that fail to be fate mapped that are from the tomato mouse, but they actually didn't get fate mapped because the efficiency is not 100%, right? Like I showed in the beginning. So we got here 2 pairs of parabiosis, and we sorted individual GCs. So we have like 4 GCs from this parabiosis pair and 2 GCs from this one. And we labeled them from GC1 to GC6. Here you can find the clonal expansion. So for example, in these GCs, we have several clones that are expanded, right? That comes from the founder cells here in orange. Here, for example, in this GC, one clone took almost like 3/4 of the GC, almost 75% of the GC, suggesting that there was a clonal, massive clonal burst happening here as well. And it was also nice to see that even on those GCs that were highly invaded, composed of invader cells that are expanded here in green, we can actually identify clones that are bursting, that are from the invader compartment, but that are capable of undergoing proliferation and getting selected. And this was also the case here in this second pair. And when we do the phylogenetic trace of these GCs, so for example, this is clone 1, this one that underwent this massive burst, and we can actually appreciate that this is the mutated ancestor. So this is the cell that joined the reaction in the beginning, the founder cell, and it underwent different clonal bursts, giving descendancy to lots of different cells. We can identify several clonal bursts, and that's what the GC should be doing, right? Selecting for cells that are most likely reacting to flu at this stage. But what was also nice to see is when you look at the invader-dominated GCs, and we see the same phenomenon happening, right? Which was quite interesting to see, which is basically those invaders that are joining the reaction, replacing the founder cells, they are also capable of undergoing clonal bursts and proliferating. So the invaders are actually contributing somehow for the ongoing germinal center reaction. And with that, we conclude that the invaders are polyclonal, they resemble founders transcriptionally, like I showed before with the SmartSeq data, and they are also capable of getting positively selected. Most likely, they are also exporting memory and the plasma cells. And if they are doing so, we wanted to understand further what are the binding characteristics of the invader cells, right? And for that, we performed parabiosis again to address what are the invaders binding to. And again, we performed the experiment, but now instead of keeping only one mouse infected like I did before, we actually infected both. So presumably, the cells that are specific for flu from a mouse could invade each other's GC. And to check for that, we performed this flow cytometric analysis at week 8 and week 12 after infection. And it was very surprising to see that when we look at the .1 mediastinal node, those germinal centers that are binding to flu, which are HA specific, they are actually coming from the .1 mouse. But those invaders, although they are here, they are never binding to flu. And the other way around is also true for the .2 mouse, where we see that the cells that bind to flu actually come from that mouse. And the invader ones coming from the .1 mouse, actually, we barely can detect them in this assay by ELISA, by flow, sorry. And the same is true if we look at a later time point, we can also see that this phenomenon is somewhat similar in a different time point. So this was quite unexpected. So it seems that the invader cells that are joining the germinal center reaction in these long-lived germinal centers, they are not binding to the antigen that gave rise to these GCs. And we also tested the same thing by Nojima cultures. Here we can assay single cells sorted invaders and the founders. Each of these lines is an invader cell or a founder cell. Here it's tomato. And we can access the binding properties of these cells for HA, NA, and NP, which are the main immunogens related to flu. And we can see that the founder cells actually bind HA, NA, and the majority bind NP, actually, but the invaders are actually not binding any of these proteins. So that really suggests that whatever these invaders are binding is not flu antigen after they join the germinal center reaction. And this can also be seen by when we sequence these invaders antibodies. Here we have some sequenced cells that we got some clonal burst points, and we assessed again by analyzing these monoclonals that we produced, and we can see that only a few of them, like IL-4 and IL-6, are actually binding flu proteins. But when we compared, when we performed this assay in a MDCK cell lysate that was infected by PR8 or uninfected, we can actually see that these clones that are binding to flu antigens, they also bound the uninfected lysate, which was very surprising to see. So when we tested in a polyreactivity panel these invader antibodies against double-stranded DNA, single-stranded DNA, insulin, which are autoantigens, and molecules like LPS, we can see that IL-4 and IL-6 are actually binding to all of these molecules, suggesting that actually these invaders that we saw binding to flu proteins, they are not specific to flu, but rather they exhibit signs of polyreactivity. So they're not truly binding to flu. And to rule out whether if the invaders are not binding to flu, do you really need to have these B cells to invade? Do they really need to bind to something? And to rule that out, we performed again the parabiosis, but now using a parabiont that was MD4 or LMP2A. So MD4 is specific for the HENAG lysozyme, which is the HEL protein. So the cells from this mouse bind to this protein specifically, or the LMP2A mouse, where the BCR was replaced by a molecule that gives tonic signals for the B cells to survive, but it's not capable of binding to antigen like a conventional BCR does. And we assessed their capacity to invade the germinal center. So when we do this experiment, we actually see that neither in the context of MD4, nor in the context of the parabiont that is LMP2A, we don't see the process of invasion happening anymore. So if you look at the frequency here, although you have chimerism in the T cell compartment and the non-GCB compartment, when you look at the invasion of the germinal center, it's not happening anymore, which suggests that the invasion process it's not happening by stochastic recruitment of the B cells that are around. They need to bind something. They need to have a BCR affinity to join the GC reaction. It's just that despite our best efforts, we couldn't really find what they bind to. So we tested for a panel of autoantigens, autoreactivity. We tested for several antigens that we have availability to, but we could never detect so far what these invaders are binding. They are just joining the germinal center reaction. They need to bind something. They need to have a functional BCR, right? So the invasion is BCR dependent, like it's shown by these experiments, but we don't really know what these invaders are binding in order to join the germinal center reaction. And furthermore, we wanted now to understand the founder's binding properties. So those very few cells, like I showed you, that resist replacement and continue on the germinal center until week 16 or 24 after infection. What are they binding to? Are they still binding to flu protein even after so much time, and that they were overwhelmingly replaced by these invader cells? And for that, we produced antibodies from these cells that resist replacement at week 16 or 24. We have several examples here by the clonal trees, like I mentioned before. And we picked these founder cells 1 to 14, and we produced their antibodies, and we assessed their binding capacities against flu protein. And we see cases, for example, the majority of them don't bind to flu anymore. So they're still in the germinal center. They were initially binding to flu antigen. That's why they joined the reaction, but they're not binding to flu antigen anymore. But we did see some cases of, for example, FO7 and FO9 that are highly binding to NP protein, which is one of the main immunogens from flu, and also FO1, which is binding to almost everything from flu. And when we tested these founder antibodies binding to autoantigens, again, double-stranded DNA, single-stranded DNA, insulin, and LPS, we can actually see that rather than being flu-specific, they are also binding to some degree all of these autoantigens, which lead us to speculate that maybe, and again, we don't have data to support this yet, but maybe one of the stay in the germinal center for a long time, like these founder cells are doing, comes with the expense of acquiring polar reactivity. And again, this is all speculation, but this is something that could be happening in this context. And to finish this up, I'm just going to show you something that we did with the collaboration from Hofbauer at the University of North Carolina. He provided us this SARS-CoV-2 that is mouse adapted. So one of the trouble that we had in the beginning was that in order to infect these mice with COVID, because we wanted to know if all this data that we are showing with flu would also be seen in the case of SARS-CoV-2 infection, right? Because of COVID and the clinical importance and everything, and to test for another kind of respiratory virus. And for that, the strains that we had at that time would require the human ACE2 mouse, and we wanted to infect the S132 tomato mouse. So it would require us to cross back to a human ACE2 in order to perform these experiments. But then Hofbauer came up with this mouse adapted strain that could actually infect wild type mice. And that's why we used it here. And so we performed the same experiments. I'm just going to go through everything that I showed you so far with flu, but basically we were capable of finding the same results for SARS-CoV-2 that we found for influenza. So the germinal centers in general, the frequency is maintained over time up to 16 weeks after infection. And we also observed the clonal replacement, the invaders coming in and kicking off the founder cells up to week 16. So the founder cells decline consistently with the data observed by flu. And when we gate on the germinal centers, for example, at week 8 after infection, we see that the invader cells are not binding to the spike tetramer, but the founder cells that are tomato positive, they are actually doing. And here in this case, there is no way of doing parabiosis in this context because SARS-CoV-2 is B-cell free, so it's impossible basically to perform these experiments B-cell free and parabiosis. And again, we show here for different mice that the founder cells are capable of binding to SARS-CoV-2, but the invaders are actually very lowly doing this job. And again, when we sequence the the PCR from these germinal center cells, we see this is 16 weeks after infection. It's a very polyclonal compartment in the invaders. We can see some degrees of clonal expansion in the founder cells. And when we sequence and look at the number of mutations in these germinal center cells, we can still appreciate the same findings that we did for flu, where the founder cells in red have a higher mutational content compared to the invaders one, and this is clearly represented here in the median of 3 different mice. So also SARS-CoV-2 infection also leads to these long-lived germinal centers that resemble the ones that we see with flu. And finally, as a last set of data, I wanted to show you this. Okay, so everything that I showed you so far was characterization of the long-lived germinal centers. So we knew that they existed, but we didn't really know how they were operating, if they were operating like canonical germinal centers, the short-lived ones, as we know. And we were able to show this process of clonal replacement that sustains these germinal centers over time. And although the invaders are coming and replacing the founder cells, they can still mutate, they can still undergo clonal bursts, and they can still get selected to survive, but we don't really know what the invaders are binding to, right? And then we came up with this question, is like, okay, so if we focus on the founder cells, those that persisted up to 24 weeks, although the minority of them, but are they still doing something productive against flu? Are they exporting memory cells that can be recalled into plasmablasts? This is very important to mention that the memory cells, once they see the antigen again, they can immediately differentiate into plasmablasts and give rise to antibody secreting cells that can quickly neutralize the antigen in a secondary exposure. So to assess that, we repeated the experiment with the tomato mouse, but now instead of fate mapping at week 2, we also fate mapped at week 12. So basically, when we put the timestamp, the tomato protein on the germinal centers at week 12, we are losing 10 weeks of export of memory cells by doing this late time point. And after doing the fate mapping early on in our control, let's say mouse at week 2, that we fate mapped the entire memory compartment, or week 12 here, we waited 4 weeks for these memory cells to be exported from week 12 to week 16. And then we boosted the footpads of these mice with 2 of the main immunogenic proteins from flu, which is HA or NP. So in the same mouse, we boosted this footpad, the left one with NP or the right one with HA. And we evaluated the plasmablast response in the popliteal nodes, which are those training the immunization here 6 days after the boost. So the question here would be like, are the memory cells generated after week 12 capable of giving rise to plasmablasts in a secondary challenge when you boost these mice against flu again? And for that, this is basically the quick experiment, the quick result, let's say, by flow. Here is evaluating, we also had naive mice as a control, but our positive control here is week 2 fate mapping, week 2 fate mapping, which are these ones. And we can see that if we fate map the entire memory compartment, of course, the plasmablasts that we see here after the recall response, they are majoritarily composed of tomato cells, which suggests that the memory cells exported after week 2, generated by flu, are capable of differentiating to plasmablasts. This is seen for NP, and this is seen for HA also. What was very nice, what was very interesting to see was that even losing the initial 10 weeks of export of these fate map tomato cells, we can still see that those cells exported after week 12, they can still get recalled and they can still give rise to this plasmablast. It was the case of 3 out of 4 mice here, the NP boost, and 2 actually, but we sequenced on this one at week 12. And when we sequence then these 4 different nodes from these 4 different mice, these 3 and this one for HA, we can appreciate that the most expanded clones, like for example, clone 1 for this mouse, clone 2 here, 3 and 4, they actually are seen as a red and a non-red version. So implying that the cells that are fate mapped here exist in the germinal center before week 12, and they were also capable of giving rise to this plasmablast. And they were the most expanded clones. And when we draw a clonal tree or phylogenetic trees from these mice, we can see in all these cases that the less evolved ones were those that were there before week 12, but those red cells exported after week 12, they're actually giving rise to more mutated cells, and that's why we are seeing them here. And one can assume, okay, so indeed, these data are suggesting that cells exported after week 12 are contributing to recall responses against flu, but how do you know if these are invader of or founder cells at week 12? Since when you post with tamoxifen at week 12, you are also fate mapping the invader ones, right? You are putting a tomato tag on the invader ones. And we really think those are founders because of the number of mutations. So if you get all of these clones, the purple ones that existed as a tomato cell and a non-tomato cell, we can clearly appreciate that the number of mutations of these cells is very high. And as I showed you before in the simple tomato experiments of the clonal replacement early on, we see that the number of mutations from the invader cells were pretty low in this level here. But in this case, the vast majority of these cells are very highly mutated, which really suggests that the founder cells actually exported after week 12 are those giving rise to the plasmablast in the recall response. So overall, these data suggest that the late founder cells export useful memory cells for recall response. And that was the end, the last chapter of this story that we published on January this year. And here you can appreciate the graphical abstract of this, where we show that, this was shown before. You have these long-lived germinal centers primed by flu or SARS-CoV-2, and we show that they go up with time and that they can still persist long after initial antigen exposure. And the reason why these long-lived germinal centers are maintained is because of this clonal replacement taking place in these structures. So you initiate the response with the founder cells that are specific to flu, but as time goes on, you see those unmutated invader clones of undefined specificity. Like I told you, we don't really know what they are binding to. We see that they are coming in and they are invading and they replace the founder cells. So in the end, you have disease composed of very few cells that were responding to flu, and all the other ones are responding to something else that we don't clearly know what they are binding to. But those few cells that resist replacement till the end, they are still capable of producing memory cells that are highly mutated, and they can give rise to plasmablast response and help in further exposures to flu. So overall, this data suggests this, we review underappreciated aspects of germinal center biology that are not clearly seen in other short-lived germinal centers, although this is a back-to-back, there's a back-to-back paper with Michel Nussensweig lab suggesting that these naive invaders are also happening to a certain extent in short-lived germinal centers primed by HIV protein, for example. But also we point out to this clonal replacement as a potential constraint to the development of high affinity antibodies in these structures. And this is something that vaccinologists might need to be aware of in order to produce better vaccination strategies. And with that, I'd just like to finish by thanking everyone that contributed to this work. So this work was performed by me, Alex Barbulescu, and also Jonathan Ershing, our beloved colleague that left us 3 years ago. So we dedicate this. Johnny was pioneering in the development of this study, contributed a lot to this study. He initiated it, and we dedicate this paper to his memory, and also people from the lab that really helped in this work, like Luca, Alvaro, Johan, other people that really made an effort to help us out to finish this story. And also people from the Musida lab, Rohan, Musida lab, Hans-Heinrich Hoffman, that helped with the SARS-CoV-2 infections in the Charles Fry's lab, our collaborators from Stanford that helped us with the autoreactivity panels, lots of supplementary data that I didn't have time to show you today. And also Ralph Parekh with the SARS-CoV-2 strain of the mouse-adapted virus, and Victoria Lab in general, Rockefeller University, Human Frontiers, and CRI for sponsoring my postdoc here in the lab. And with that, I'd like to finish this. Thank you for your attention, and I'm open to your questions. Thank you.
Thank you so much, Renan. It was a terrific presentation, amazing results you have. So we are now open to questions. If anyone have any questions, you can raise your hands or put it in the chat. So
should I stop sharing, Luisa?
Yes, please, Renan.
Okay.
Miriam, do you want to start in the EU side?
Yes, I think I have a question. It was really, really interesting. I think I'm going to just turn on the lights because it's a bit dark. Okay, see, can you hear me well? I thought the mic was.
Yes.
Yes, okay. So it's a very, very interesting question. I'm gonna start just to break the ice. So one question I had was based on some work from a while back, talking about how the selection on the colleges could happen due to QA1 expression.
Miriam.
I'm not
Miriam, sorry, we are just seeing your back. You are not. The cameras are not capturing you in the room. It's just the Christian computer. You can sit in front of the computer.
It's here.
Yes, now we can see you. Yes, the cameras, they are not working well.
The sound is over there?
But the microphone is over there, and
yes, but we can hear it very well.
It's great.
Okay. See if you can hear me well, then I'll stay here.
Yes.
Try to speak a little bit louder. I'll do my professional voice. So the question will be, there's some work from a while back now talking about how the QA1 CD8 T cell, restricted CD8 T cells can change the tolerance or the response of B cells indirectly, like killing CD4 and helping CD4 T cells. And what I wonder is if you think that could play a role, considering that you see mostly viral infection and that, let's say, cutting of the repertoire may help on the disappearance of the founders. And then therefore, but maybe because of persistence of or limit function of the viral infection, then you still have some reaction happening, and then you have the entrance of the invader.
Okay, so it was failing to some extent when I was trying to hear you. Let me see if I understood your question, Miriam. So you were asking me if about the role of the T cells, CD8s and everything in this context of long-lived germinal centers, and if persistent infection or acute infection would somehow affect the behavior of the GCs, is it?
Yeah, something like that. If you have to have like some lytic effects that would release self-antigens, and that's why you have a different repertoire from the later germinal center versus the beginning one. And if this tuning of the follicular T cells could be at play in that phenomenon.
Right. So yeah, so one of the things that we didn't deeply investigate in this context of long-lived germinal centers is the TFH compartment. And this is a question that we always get, and we didn't really very close to it, but it remains to be addressed. What I can tell you from some supplementary data that we have, this was also published in Ziv Schumann's paper in 2016, the science paper with Gabriel, was that the TFH compartment has an exchange capacity that is way better than the B cell ones. So we think, we show that actually, that the TFHs are invading way more these germinal centers than the B cells to some extent. So there is indeed something happening to the TFH clonal compartment. So we don't really think that the same TFHs that see that the reactions, let's say the founder ones, are the same resisting to the end. We think that they are much more exchangeable compared to the B cell ones. That's all I can say about it now. We never sequenced the TFHs and did clonal analysis that like we did with the B cells in these germinal centers. And we don't really know what are what are these TFHs eventually binding to, right? Especially late after infection. And the other question was, so one of the things that we notice, and this is something very interesting to some extent, is that, you know, in all of these experiments that we did with flu, like I said, flu is cleared after a week of infection. So presumably, let's say 3 or 4 weeks after infection, you don't have antigen in the system anymore, or they are like almost entirely cleared, right? Although we don't really know if there is some deposits of antigens in the germinal centers that will respond to it. We can't really track the antigens over time. But we actually can now with a new mouse model we are making, but we never did this experiment. But one of the things that we see is like when you have a chronic infection, for example, the LCMV1 or Plasmodium schabauri, which like remains in the bone marrow even after getting cleared from the blood, remains in the bone marrow, we still see these long-lived germinal center structures. So we don't really think this is particular to chronic or acute infections. We see these long-lived germinal centers in both cases, depending on the infection. So we really think that, you know, it's something else that's driving these germinal centers. Our best hypothesis about these germinal centers is, and this is based on several different observations that we have from other models as well, but we think it has to do with the specific lymph nodes. So those nodes that are constantly exposed to environmental antigens, like the mediastinal node, the mouse is breathing, right? So there's always like antigen being deposited into the mediastinal node, the portal node that's draining the liver and the blood, the mesenteric nodes that are draining the gut, which is exposed to autoantigens, you know, food and microbiota and everything. Those lymph nodes, they are more likely to form these long-lived germinal centers. And what we really think that is happening on the case of the mediastinal one that we studied is that flu acts as a priming agent to induce germinal centers. But as time goes on and flu gets cleared, the invaders that join the reaction are those that are responding to environmental antigens that are depositing there. So there is no reason to dissolve the whole structure every time and start a new germinal center reaction. This could be energetically expensive for the body. So, you know, you have the scaffold, you have the structure of the germinal center that was seeded by flu in this case, and then you have the deposit of these antigens over time in the germinal centers, and the invader cells that are getting there are binding to environmental or antigens that's coming from the air, airborne antigens. And this is also the case that we observed in a previous paper from the lab from Carl and Ozad in Nature in 2020 as well, about the gut draining germinal centers that persist also for a long time. So it's like you trigger the germinal center, but there is no need to dissolve it and, you know, start all over again. You just keep seeding those cells, and in the end, you have these long-lived germinal centers that are not specific to flu anymore, but they are fighting, you know, high number of different antigens that are taking place in the same structure. So that's more likely. Again, this is also speculation. We don't really have data to pinpoint this yet, but this is like one of the things that we observe. When you look at the popliteal node, for example, that's behind the knee, right? It's way more difficult to induce long-lived germinal centers there because it's not constantly draining anything, right? So I'm just like trying to connect these points to make this point that this could be a scenario to explain the persistence of these germinal centers. But again, not enough data yet.
In that sense, do you think that have you tried to model? I saw your model in the beginning where if you use the free antigen or the antigen and the infection, right? And have you tried looking at the popliteal, for instance, where you basically do another injection before you actually had a cut down, an expected cut down from the first one, to check either using the same antigen or another antigen to see the germinal centers just be repopulated with the new specificity, or try to model exactly what you said, but in a lymph node that shouldn't react that same way?
Yeah, that's a good experiment. We never really tried that specific way that you were saying. But for example, if you get naive mice and you prime them with HA or NP, just like I did in the first experiment, but no previous exposure to flu, you don't really see the germinal center lasting for more than 4 weeks. So 4 weeks is the maximum that it lasts, right? So, if eventually another experiment that we could do is to try to get these HA and MP and do an intranasal administration and see if we can prime long-lived germinal centers with these antigens intranasally, because then the mediastinum is the one training, right? Or on the other hand, we could try to inject flu in the footbed and see if it doesn't induce, you might provoke a germinal center reaction, but maybe it won't be sustained for a long time, right? So there are all these phenomena that we think could be happening. And one of the things that we tried actually was we made some new monoclonal mice that are, BCR specific to flu, to the head of the HA protein. And we transferred these cells to wild type mice and induced a germinal center using these monoclonal specific cells. And what we see is that if the germinal center is initiated by monoclonal cells, they die within 2 weeks. So you really need to have a polyclonal compartment taking place, in order to sustain this germinal center reaction. So these are like observations that we have that suggest that, these cells have to invade in different specificities. They need to sustain with specific B cells for flu, but those that join later, unlikely to bind to flu, but they need to bind something. And the best bet that we have is the environmental antigen so far. And importantly, maybe someone is going to ask that, but we also performed these experiments in germ-free mice, and they still induce long-lived germinal centers. So it's not microbiota driven, apparently.
Okay, yeah, that wasn't my list. I was going to leave it for later.
Yeah, because that's normally a follow-up question from all of these.
It's true. Lisa, we have another question over here. If you don't have any there, and just to show a little bit of my ignorance, when you show the labeling to show the germinal center B cell, then you show FAS. I was wondering, is it fatty acid synthase or is it the FAS from the FAS ligand molecule?
It's CD95.
Okay.
CD95, yeah. So one of, there's a lot of apoptosis taking place in the germinal center, and it's mediated by FAS ligand, right? So 99% of these cells are going to die because, like I said, the AID mutations are random, and it's unlikely to induce higher affinity antibodies. So those that decrease affinity, they're going to be eliminated by apoptosis. So it's CD95 pathway that makes these cells to die. Yeah.
Okay, thank you. Okay, I'm just gonna try to make the microphone better. Maybe now. Do you hear us a little bit better?
Yeah.
Yeah, very well.
Yeah.
Mm-hmm.
Okay. So Lucas Bonini has a question, our PhD student in the program.
Hi, Lucas. How are you?
I'm fine, thanks. Thank you for your talk. It's very interesting data. I was wondering, because you showed in the end a boost with the antigens, right? What do you think would happen if, let's say, because we get exposed to flu all the time, and of course we are exposed to different strains of flu all the time. So what do you think would happen to those long-lived germinal centers if you reinfect the mice with a different strain, not just like a boost with the antigen, but they actually get exposed with a different strain of flu? Do you think the new flu-specific clones of B cells would take over those germinal centers, or do you think new long-lived germinal centers would be formed?
That's a very good question, Lucas. We haven't tried the double infection with different strains to assess exactly this, but if by what we learned from these long-lived germinal centers, and if the hypothesis that you have constant antigen deposition from environmental, from the environment into these long-lived structures is correct, I would presume that when you come with a second strain of flu, you would have invaders that are binding to the second strain of flu to join the germinal centers and eventually cross-react to the previous strain. So that's my assumption, although I don't have data to show that yet. Because, my impression is like, whatever you give, like in the case of in our system, we just prime it with flu, right? And the invaders are binding to something and going in. But whatever you immunize later on in the mediastinal node, we will drive those cells to go. So let's say if you give NP over intranasally or you give flu, I would guess that the invaders that join the germinal center reaction would be specific for the immunizing that you are using. That's the presumption.
And you just a small question, because I don't think I remember you talking about it. And you don't see those germinal centers being formed in very distal lymph nodes, or like maybe in the cervical lymph node or whatever, that is very far from action.
Yeah, so this is a good point, because actually this was published in Terry Aldo's paper in Cell Reports 2 years ago, where he also showed the germinal centers induced by flu. And you do have germinal center formation in the spleen of these mice. What I'm not sure about, we never really looked at the spleen germinal centers and their overall contribution to the responses, but I think it was somewhat long-lived as well in the spleen, not as much as the ones in the mediastinal node, but they persisted for some time. In terms of magnitude, the spleen germinal centers responding to flu are not as big as those from the mediastinal, but you have a local response. So that would imply that you have some draining. Yeah, you have some viral particles going to the blood and draining to the spleen, and that's why we are having the response over there as well. So it's not only, basically answering to your question, it's not only the mediastinal node. The spleen also forms germinal center against the flu in this case.
Okay, thank you.
So I have a question. Can I ask? So Renan, very beautiful work, man. I kept taking notes of questions here, and you kept answering them along the presentation. So I thought about a question, and actually you answered it when you were answering me. I thought about the availability of flu antigens in the follicular dendritic cells, and you said that you did not really measure that, right? Because when you have the invaders coming on, you probably don't have flu antigens anymore. You may have environmental antigens or autoantigens. So I was thinking about
Correct.
These long-lived germinal centers. Can they be a potential driver of autoimmunity then? What could be the drivers? Not the drivers, but the regulators that would keep those autoimmune T cells in check when you have. I think the same thing you said. You have a primed environment. Whatever comes is going to be presented and generate a stronger immune response, right? So what will keep those autoimmune cells in check? Are you studying that? If T follicular regulatory cells play a role, or the bone marrow, or whatever tissue,
Yeah, so Diego, it's a very good question, and it's a tricky one because it's hard to explain. But what I can tell you, it totally makes sense the way you're thinking about it, because if we do show that those late invaders and the late founder cells get some polyreactivity characteristics that enable them to bind to double-stranded DNA and to insulin, and then people start to think about type 1 diabetes or lupus or things like that, right? So it totally makes sense that the way that you're thinking. What I can tell you from our, let's say, experimental evidence is that even following these mice, like until they die after flu, like I said, these germinal centers persist for 6 months at least. And I can tell you, this is what we showed in the paper, but even a year after infection, these GCs are still there, So it's like half of a mouse lifespan, right? A mouse should be living, let's say, on average 2 years. So when we follow these mice up to 2 years until they die, they never exhibit any signs of disease or, autoimmunity or things like that. We didn't really do a very deep analysis to really measure, let's say, antibodies against autoproteins and everything, but they seem to be fine. They seem to be happy. So I don't really think that these germinal centers are inducers of autoimmunity, especially because, when you go to the clinic and you go to human data, I mean, these long-lived germinal centers are happening in the gut all the time, right? So the gut is the best example one can ever have because you are constantly exposed. They are long-lived forever. We have responses ongoing against microbiota, oral proteins coming from food and everything. And we don't really have cases. Otherwise, I mean, we would all be have autoimmune diseases, right? Because of these gut germinal centers. So I don't really think there is substantial clinical evidence to suggest that, although I wouldn't really say it would be impossible in certain, contexts to have this. And one thing that we have to think about is that polyreactivity is not the same as being high affinity for double-stranded DNA, for example. So it's like, maybe we don't really know the affinity, at least from the cells that we produce the antibodies from. They don't really seem to be binding very strikingly compared to our control antibodies, for example. So I'm not really sure if they would be able to outnumber or to, these cells would be exported in a significant number to induce autoimmunity and everything, and to break secondary tolerance, right? So even though you have these properties going on, let's say if one or two or a few cells escape from these long-lived germinal centers and eventually producing antibodies against these autoantigens, you still have secondary mechanisms to control it. So I don't think they really overwhelm the system in order to provoke autoimmunity. But again, based on everything that I told you now, and from the human evidence and thinking about the gut germinal centers, which are crazy high, right, and crazy long-lived, I don't really see an evidence for that. That's all. Yeah, I know it's sort of a speculative question, but when I stop to think about this, that's all I can think about.
But then you think that the major regulatory mechanism there would be, I was thinking about that. Nobody gets autoimmune just because they're reacting to some antigen, some infection. But so you did not measure the affinity of these autoantibodies if they get, for example, one of the secondary ways to control autoimmunity is exactly this. You don't have enough affinity to keep those B cells active, right? They die by apoptosis later and everything. I think mainly the peripheral mechanism would be the main mechanism to control the autoimmunity, the potential autoimmunity that could be generated.
Yeah, exactly. And I mean, we do have these affinity measurements, but like I said, it's nothing compared to our control antibodies. They don't seem to be binding to the same affinity. And I think it's also a numbers game, we don't really see, when you think about autoimmunity and all those B cells producing antibodies, you think about those big nodes composed with a single clone, that there's a massive, it's like a tumor and lots of cells, and we don't really see this in this mediastinal node. So it doesn't seem to be pathogenic at all. That's what I'm saying. Yeah.
Yes, congratulations for your very nice data.
Thank you for your questions. Yeah.
Renan, just out of curiosity, I was thinking about even if the antigen goes out after a few days or weeks after the injury, it's more a curiosity than a question, but for how long a follicular dendritic cell can retain the antigen in the membranes, and how does it can impact in the generation of these long-lasting germinal centers?
Yeah, that's a very good question. I don't really know if that was evaluated before, how long a follicular dendritic cell can hold an antigen. I think it has to do with the draining, right? As long as you have draining of the antigen to the germinal center, to the secondary lymphoid organ, this gets deposited on the top of these FDCs, right? And in the case of the mediastinal node, this is happening all the time. Like I said, it's always draining environmental antigens that deposit into the lungs and everything. I don't really know about the turnover, how long it takes for the antigen to be cleared. So one of the factors that might affect this is the constant draining, and also the the speed velocity that this antigen is taken by B cells from these FDCs, right? Because the FDCs is supposed to be like a tower of antigen. Maybe it does something else, but that the B cells come, and once they have high affinity, they come and they dispute with each other to get the antigen and to present the TFHs, right? And eventually get selected. But I don't really know about the turnover. Yeah, and this is difficult, Luisa, because it's very hard to track the antigen into the germinal centers. You know, like we can stain for the FDCs, like I showed by CD35 IV. We can stain for any kind of cell that you want to see in the germinal center, but tracking molecules into the germinal center is very difficult. So we don't. What we are trying to do now is an indirect system to evaluate this antigen persistence. Like I said, we have these monoclonal flu cells for HA, and an experiment that we are willing to do that we haven't done yet, but we are willing to do, is to repeat the infection with B6 mice, a very simple experiment. We infect B6 mice with flu, and several time points later, we transfer these monoclonal cells, especially late after infection, to these infected B6 mice. So one would assume that if the antigen is still present into the germinal centers at late time points, those cells specific for flu should join the germinal center reaction. So that's how we test it. So let's say if I transfer, a positive control will be transferring, infecting now, and 3 weeks later we transfer the flu-specific cells. Those should join the reaction because it's still early. But 12 weeks after infection, if they don't join the GC reaction anymore, that would assume that the antigen is not there anymore. So that's an indirect manner to test antigen availability in these GCs, especially at late times after infection. But like the antigen itself in the GCs, it's very hard to measure.
Thank you, Renan.
Follow up on that, would you have to irradiate the mice, Renan? Because for T cells, I guess you would, but I'm not sure if for B cells you would.
You are asking about this specific experiment that I just described?
Yeah.
No, you don't need to, because you are basically testing, I mean, these cells that you're transferring are of very high affinity for flu. So if the antigen is there, they will find their way through. They will join the reaction. So actually, it's a good point. Maybe we should have different readouts, because when a B cell that has high affinity for an antigen sees the antigen and binds to it, they can either join the GC reaction, that would be the first readout, so we could evaluate that. The second readout would be proliferation, so we could post these transferred cells with CFSE and see if they proliferate once you transfer. And the third readout, which is the most likely that we see in some systems, is the immediate differentiation into plasmablasts. So those high affinity cells, instead of joining the GC reaction, they can give rise to extrafollicular plasmablasts, like I showed in my first slide. So either of these could happen and would be point out to antigen present in the node, right, in the germinal center. But yeah, any of them could be observed. But you don't need to irradiate because of the very high affinity. That's the differential, right? Because if you transfer wild type cells, that's more complicated. But when you transfer very high affinity, they should join or do something when they see the antigen.
I think we have asked all the questions over here. Renan, due to the time, I will skip the rest of my list. But thank you very much, and thank you, Luisa, for inviting Renan to talk to us. It's very interesting work. Congratulations.
Thank you so much, Renan, again.